Okay, so it's my great privilege, I shall just bring up the presentation, to invite our first speaker, Professor David Wraith. David's been working in immunology for a couple of years and has a bit of experience in the field. So particularly with regards to looking at tolerance, induction, particularly in focusing on the area of peptides in regulating autoimmune diseases. So it's a great pleasure to have David, who has two hats on. He's a Professor of Immunology in the University of Bristol. He's also involved in Epitopes, which is a company based in both, as a spin-out company from the University of Bristol. And I'll let David tell you a bit more information about that. So I'll just bring up his slides.
Okay, well, thank you very much, Jeremy. Thank you for the kind invitation to come and talk at this meeting, and thank you all for coming. Now, what I would like to do today is talk about the work that we're doing in trying to oppose the immunogenic properties of various proteins through the design and development of tolerogenic T-cell epitopes. And before saying any further, as Jeremy pointed out, I'm CSO at Epitope International. This company designs and tests peptides for antigen-specific immunotherapy. And I just wanted to point out that at the end of the talk, I'm going to be talking about a recent clinical trial which has just completed. And this is the disclaimer for that part of the talk. So what do we mean by unwanted immunogenicity? Well, there is, of course, the immune pathology that arises with infectious diseases. This is one of the consequences of infectious diseases, which can cause more disease, in fact, than the infectious agent itself. So immune pathology itself can be an unwanted immunogenic property of infectious agents. But I think these are the areas that most of us would think of in terms of unwanted immunogenicity. Allergic responses, autoimmune diseases, hypersensitivity reactions to self-antigens, transplant rejection, and of course unwanted immunogenicity towards medical products, drugs such as penicillin or biologicals such as monoclonal antibodies. And I'm going to be touching on some of our work on factor VIII in terms of the biologicals against which there are or there is unwanted immunogenicity. So dealing with this, well, I think everybody in this room would accept completely that at the center of the adaptive immune system lie CD4 T-cells. CD4 T-cells control the adaptive immune response to both self and foreign antigens. And as such, our aim is to overcome unwanted immunogenicity by targeting specific CD4 T-cells with specific antigens, okay? And to do this, we design and develop tolerogenic CD4 T-cell epitopes and administer those in order to overcome the unwanted immunogenicity. Right? So why would we want to be antigen specific here? Well, I think this pyramid here, this figure here, just emphasizes the experience that we've had in developing treatments for multiple sclerosis in particular. And I know Florian is going to be talking specifically about beta interferon, so I won't mention that. But the aim of this slide is to emphasize that we aim to produce more specific, disease-specific treatments in order to reduce the frequency of side effects. As I mentioned, Florian will be talking about beta interferon. Campath-1H is an antibody that depletes all leukocytes in the body and is a very, very effective way of stopping multiple sclerosis, certainly at the relapsing remitting stage, directly in its tracks. However, that treatment is associated with about a 30% incidence of Graves' disease in the treated patients. And I think we would all accept that that is not highly acceptable. We can block T-cell migration with antibodies against molecules such as VLA4, but in doing this, we prevent immune surveillance in the brain, and this allows certain infections, particularly the JHM virus infection, coronavirus infection, to crop up in a small number, but probably an unacceptable number of individuals. And then we have the more antigen-specific approaches, including the random polypeptide copaxone or glatiramer acetate, which is a frontline treatment for MS currently. And I'll be talking about our antigen-based approaches in a moment. So a lot of people say to me, well, this antigen-specific approach moves us away from non-specific anti-inflammatory drugs, and it's a totally novel concept. Well, it's not. Antigen-specific immunotherapy has been tried and tested, has been in the clinic for over 100 years, okay? And this is one of the early reports of this approach from Freeman and Noon in London. And I think you would all agree that if we could describe a new treatment for a disease such as multiple sclerosis in these terms, possibly not quite so old-fashioned as this, in that even in those days they didn't have a word for allergy. They used to call it a constitutional disturbance, which I think is a rather nice phrase. But if we could describe a treatment for multiple sclerosis in these terms, we would be very pleased. Okay? Now, I've got a series of questions that I want to address with you. The first one relates to the nature of the antigen. And one of the problems with antigen-specific immunotherapy, and one of the reasons that it hasn't really caught on for treatment of autoimmune diseases, is because there are complications of using intact antigen. Of course, intact antigens can cross-link IgE, hence potentially causing anaphylaxis. They can stimulate generation of antibodies. Claude Chenain, some years ago, showed that treating a primate model of multiple sclerosis with the protein myelin oligodendrocyte glycoprotein led to an enhancement of disease through the induction of antibodies. And Len Harrison and his group showed that oral administration of intact antigen could potentially stimulate CD8 T-cells. So the argument, the logic, was then to move away from using intact antigen and instead to focus on CD4 T-cell epitopes. And this is what my lab and Epitope has been doing for many years now. So it all started back in the late 1980s and led to an early publication showing that administration of peptide epitopes would prevent autoimmune encephalomyelitis the model of multiple sclerosis. And this was important because this showed that administration of one epitope was sufficient to prevent disease induced with spinal cord homogenate, which is actually virtually pure myelin, because the spinal cord is mainly white matter. So this treatment with one epitope, one peptide, is sufficient to downregulate the response to whole myelin. Subsequently, we showed that the approach could be used for treatment of a model of allergy, in this case house dust mite allergy, using the antigen DERP1. And if I just run through these slides very quickly, what this showed was evidence of the phenomenon called linked suppression, whereby administration of one epitope, if you look at the 111 to 139 epitope here, this epitope is able to suppress the response to the whole allergen, the response to itself, the response to peptide 780 to 100, and the response to peptide 21 to 49. So all of the epitopes, all of the responses to all of the epitopes within the protein are being suppressed by administration of one T-cell epitope alone. This is the phenomenon of linked suppression. Okay? The second question, which is a very important one, relates to peptide design. And Steve Anderson, who worked in the lab at the time, was trying to solve a mystery, and this was why one particular T-cell epitope from myelin basic protein failed to induce tolerance. Okay? And he showed that not all T-cell epitopes induce tolerance, and the reason is because if you look at the MHC class II molecule, it's a relatively open cleft. Okay? And Steve discovered that when you add peptides from outside of an antigen presenting cell, or outside even the body, those peptides can bind to the MHC in a distinct conformation compared to the way that peptides are generated during processing of the protein inside the cell. Okay? So you end up with a distinct conformation of peptide in the cleft, and that peptide fails to induce tolerance to the naturally processed epitope. So the rule is that peptides must be designed to mimic the naturally processed epitope. So we design our peptides to function as antigen processing independent epitopes or apoptopes, so that they bind directly to naked MHC on antigen presenting cells. And the truth is that immature dendritic cells in the body have a high proportion of empty MHC at their cell surface. So hence, by giving soluble peptides, you actually target the immature dendritic cells, which give tolerogenic signals to the immune system. And in fact, this approach is highly effective as a means of treating a wide, varied range of autoimmune diseases. These are all models of autoimmune disease, and I'd like to emphasize two studies here. One from Steve Anderson again, who showed that administration of one epitope from myelin would induce bystander suppression. That epitope would suppress the response to other epitopes within myelin. And also these three studies from different groups working on the non-obese diabetic mouse, where distinct epitopes from different proteins would all suppress the same disease. Again, evidence of bystander suppression. So we have very good evidence that peptides can suppress autoimmune conditions, that they can induce linked immune suppression and bystander suppression. So the obvious question is, well, how does it work? The first possibility is that peptide administration induces cell death. And clearly this can happen. However, if we were inducing cell death among all antigen-specific CD4 T-cells, it would be very difficult to explain how linked and bystander suppression function, because these are active processes. We can be inducing allergy. Again, the question is whether anergic cells can mediate linked and bystander suppression. Finally, are we modifying the function of cells so that they're now capable of inducing linked and bystander suppression? Now, much of this, or the mechanism of this, has been worked out using animal models, and I completely defend their use, because without the use of these animal models we couldn't work out the mechanisms involved, and without knowing the mechanisms involved it would be virtually impossible to take this approach through into the clinic. So if you take this model of multiple sclerosis and give soluble peptide 2 times per week, this totally prevents induction of disease, and most importantly, the induced tolerance is reversed by administration of antibodies to IL-10. So in this case, interleukin 10 is responsible for the immune suppression that we're measuring. And then you ask, well, which cells is the interleukin 10 coming from? And as you all know probably, interleukin 10 can be made by virtually all cells of the immune system. And surprisingly, you might think, this does not require the presence of FOXP3 regulatory T-cells. So the interleukin 10 is not coming from what you might consider to be the most likely cell involved, the FOXP3 positive regulatory T-cell. It is in fact being induced, in this case, in Th1 cells that downregulate inflammatory cytokine secretion and upregulate secretion of interleukin 10. What I'm talking about here is the ability of soluble antigen administration, soluble peptide administration, to elicit a classical negative feedback loop. So treating Th1 cells drives them to produce interleukin 10. The interleukin 10 then feeds back on antigen-presenting cells, downregulating expression of the co-stimulatory molecules CD80 and 86, downregulating MHC class II, but most importantly of all, blocking IL-12 secretion. These cells are then no longer capable of stimulating differentiation of any further Th1 cells, right? So this, in fact, explains the process of linked and bystander suppression. Because, of course, during an immune response, we might expect these dendritic cells to be presenting all of the epitopes within a given antigen, but potentially also the antigens within a targeted tissue in an inflammatory condition. Now, we speculated a few years ago that there might be mechanisms whereby other T-cell subsets also mediate negative feedback regulation through the same process. Howard Weiner here in Boston and his group suggested that this was true for Th17 cells. And more recently, Cook, Goodenow and Altin in Australia have described a what they call an inhibitory Th2 population, where IL-10 is increased, cytokines such as 4, 5 and 13 are suppressed, and they argue that this is an inbuilt activation-induced feedback inhibition mechanism against excessive or chronic Th2 responses, very much mirroring the process that I've just described for Th1 responses. So this is the mechanism, and it applies both to Th1 and Th2 immune responses. So now a little bit about designing tolerogenic peptides. And these are the various different processes that we go through at Epitope for selection of tolerogenic epitopes. We do in silico computer-based MHC peptide binding analysis using various different algorithms. We do in vitro biochemical peptide binding assays. We work with pro-immune to do peptide binding analysis. Most of the work is focused on ex vivo epitope identification, both using HLA transgenics and human T-cell samples. And finally, we can modify peptides to optimize them, particularly for solubility. The first example that I wanted to discuss with you, as I mentioned, was factor VIII. And the problem with factor VIII administration to haemophiliacs is that it induces antibodies, measurable antibodies, in about a third of all individuals. And this is particularly important and particularly dangerous in haemophiliacs with the inversion mutation who don't make any effective factor VIII of their own. And of course they recognize the administered factor VIII as a foreign protein. Okay? They make antibodies very rapidly, resulting in joint bleeding and other complications, often resulting in death. This is not a terribly uncommon condition, and it's currently managed by infusions of increasing doses of factor VIII, and treating the inhibitor patients can be a major burden for health services. High dose tolerance induction is tried, is often successful, but it fails in about 30% of individuals and is extremely expensive. Finally, bypass reagents such as factor VII can be used, and again these are very expensive. So what we have done is to work on an HLA transgenic factor VIII knockout using the B1-1501 DR2 molecule as a target antigen, and designed a set of peptides which by in silico analysis were designed to be high affinity MHC binding epitopes. Okay? These were then placed in the pro-immune reveal analysis, and this is the analysis that was done by colleagues at Baxter, Kathy Steinitz, and Birgit Rapert, and these two epitopes come up in this analysis as high affinity MHC binding epitopes. And most importantly, you'll notice the red here, if I remember correctly, is the B11501 molecule. So these are all, or the majority of these are B11501 binding epitopes. These two are clearly binding promiscuously to various different HLA-DR molecules. This is one example where the in silico prediction very much is reproduced in the biochemical analysis of peptide binding. We then immunized and selected out T-cell hybridomas from this model of hemophilia, and taking the hybridomas, we were able to confirm that the majority of these hybridomas now recognize these two predicted T-cell epitopes. Okay? And they function as epitopes, as shown by the fact that the intact protein factor VIII is presented by antigen presenting cells that are live, but is not presented by fixed antigen presenting cells, whereas the epitopes themselves, the T-cell epitopes themselves, are presented by both live and fixed antigen presenting cells. So this means that these epitopes function as antigen processing independent epitopes, or epitopes. Okay? So we took these peptides into an experiment to test whether they would induce suppression of the immune response to factor VIII. A dose escalation is done here, treating and then giving the higher dose of the peptide mixture, the combination of the two peptides that I mentioned to you. Okay? We then immunize by giving factor VIII as you would give it to a haemophiliac, either intravenously or in this case subcutaneously. It doesn't matter, you induce anti-factor VIII antibodies either by the intravenous or subcutaneous route, and then you ask, how does that affect antibody production? And of course, for factor VIII, it's a clotting protein. The biologically relevant antibodies are those that compete or that interfere with clotting. And this is the result. So treatment with this mixture of peptides here virtually totally blocks the generation of neutralizing antibodies. It doesn't prevent generation of all antibodies, however, it blocks the neutralizing antibodies. Okay? So I think to conclude from this part of the talk, for this which is a foreign antigen, effectively in this model, peptide binding, peptide MHC prediction and peptide MHC binding is an informative tool for therapeutic peptide design. And as we see, treatment with soluble derivatives of the dominant T-cell epitopes achieves highly effective antigen-specific immunotherapy. Now, I'd just like to move on to talk about our work on multiple sclerosis. I'm sure you all know about multiple sclerosis. It tends to start out as a relapsing disease, and in many individuals this progresses into a secondary progressive phase, a more chronic phase of disease. The pathology is now quite clear. There is a perivascular infiltration into the CNS as lymphocytes and monocytes cross the blood-brain barrier. Once they infiltrate the blood-brain barrier, they produce demyelination, where particularly the monocytes, the activated microglia and macrophages strip away the myelin sheath from nerve axons. And this results in so-called axonal transection, where the axons swell up and the axons become divided and no longer functional. You can see microglia and macrophages engorged with lipid from the myelin at the site of inflammation. And the demyelination, this is a Luxol fast blue staining of an MS plaque, reveals large areas of demyelination. And within these areas of demyelination there is a high degree, in these so-called chronic plaques, there's a very high degree of axonal transection, and eventually the gliosis that results produces the so-called fibrotic plaques. So this, of course, for antigen-specific immunotherapy is a much taller order, because even to this day we're not absolutely certain what all of the full range of antigens are that are recognized in MS. Here is the nerve axon, here is the oligodendrocyte producing the myelin sheath around the nerve axon. And if you look into the myelin sheath, there are three major antigens of immunological interest: myelin basic protein, myelin oligodendrocyte glycoprotein, and proteolipid protein. And we at Epitope have identified epitopes from within all of these proteins, starting out with myelin basic protein. And if you look across myelin basic protein, there are some 8 regions of immunological interest, in other words T-cell epitopes. And from these, the terracotta ones here are minor epitopes, infrequently recognized by MS patients. And these 5 here are more frequently recognized. Interestingly, this one at the end here defied design of an epitope, but we were able to design epitopes from these 4 regions, and these are known as MS1467. And you'll notice here that there are 2 HLA-DQ6 restricted T-cell epitopes and 2 DRB11501 restricted T-cell epitopes. And the reason I'm telling you that, and I'd like you to remember that, will become important in a moment. MS1467 is a mixture of these peptides and a mixture of equal parts. Okay? Now we subjected these peptides to the Reveal MHC binding assay at pro-immune, and the results are absolutely fascinating. Because if you look at the 2 DQ binding epitopes, neither of those give a signal on the HLA type that we know these epitopes are restricted by. If you look at MS6, this is a B11501 epitope, and again there is no signal on this assay. MS7 is different. So what is it about MS7? Well, why is it that this protein appears to be generating a T-cell repertoire of peptides that have very low affinity for MHC? Well, the answer is that myelin basic protein is expressed in the thymus and influences T-cell selection. Okay? The T-cell repertoire coming out of the thymus will be deleted or depleted of those cells that see T-cell epitopes with high affinity. This was shown many years ago by my lab and subsequently by Joan Goverman and Paul Lehman in 2 different models. And I believe it's absolutely the case that high affinity epitopes from myelin basic protein will induce deletion. Now, how then do you explain MS7? This is a reasonably high affinity binding peptide, and yet there are still T cells escaping tolerance. Well, it turns out, as we then showed with Colin Watts in Dundee, there is an enzyme in the thymus called asparagine endopeptidase that cleaves this epitope bang in the middle, right? So in fact this epitope is destroyed in the thymus, hence allowing T cells specific for it to escape deletion, right? And that explains why this can bind so well to the MHC. So on to the important stuff, that is, does it work? And we first wish to test this out in as many preclinical models as possible, and we thought the most relevant would be the model developed by Lars Fugger and colleagues in Oxford, or in Denmark and then Oxford. This mouse expresses HLA, DRB11501, and human T-cell receptor specific for myelin basic protein. And if you treat with peptide, you reduce cell proliferation, prevent or suppress interferon gamma, IL-17 and GM-CSF secretion. And treating with peptide after induction of disease with myelin, in male and female mice you virtually completely block induction of disease. Most importantly, if you start treatment after disease onset, you see a dose-dependent suppression of disease. And this is giving peptide twice per week, starting after disease onset. So even that first dose of peptide is having an immediate effect on ongoing disease. This dose of peptide is quite similar to the dose effectively that we then took into the clinic for treatment of patients. This is important. This is comparing Copaxone glatiramer acetate with MS1467 starting after disease onset. And unfortunately the glatiramer acetate giving 2 milligrams per day did not have a significant effect on disease. This is work conducted by our colleagues at Merck Serono, and they were looking for a suitable model to use as a relevant species for tox studies. And ATXMS1467 induces disease in the Lewis rat if given in adjuvant with pertussis toxin, and you can suppress disease by giving the peptide in soluble form. And under those conditions, yet again, you suppress both Th1 and Th17 cytokine responses. So we took this initially a few years ago into a clinical trial in secondary progressive patients, giving, as you would expect, a dose escalation up to 800 micrograms of peptide. Primary outcome was safety and tolerability. We were able to measure immunological parameters as well. And in this trial, there was no selection of HLA type, and as you might predict, no effect on clinical outcome. These patients were all severely affected. There was one example of a patient who had optic neuritis, whose visual acuity improved dramatically throughout the course of the trial, but we have to state that this is anecdotal at this stage. Looking at the immunological parameters, we had to measure immune responses to vaccine components. The PPD, as you all know, is a component of the BCG vaccine, and there was no significant change over the course of the trial in the response to PPD. However, in those patients who had a significant response to myelin-basic protein, they all showed a downward trend. And most importantly, if you then compared 3 months after the end of the trial, the immune response begins to come back again. So on the strength of this, we moved from the secondary progressive stage into early onset relapsing remitting disease, and undertook an extensive trial in 43 patients, giving either intradermal or subcutaneous treatment, extending the high dose treatment for longer. And what I'm going to show you now are results from the Intradermal Group, looking at the MRI scan from this group of patients. And this is the period during which the dose escalation is occurring, and this is the period during which the patients are receiving the high dose of treatment, and this is producing a 78% reduction in gadolinium enhancing MRI activity in these patients. This appears to persist for about a month, and then subsequently the MRI activity increases once again. So this implies that this and the previous analysis implies that for effective suppression of disease, one would need to continue treatment in this way. Of course, the major point about all of these trials is to measure safety, and effectively we've now looked at 49 patients, and there is no evidence of any unexpected safety signals, with the majority of adverse events relating to MS activity in some patients and some minor injection site reactions. So I think what this tells us in terms of peptide design is that for foreign antigens, MHC peptide binding is an informative tool for and treatment with soluble peptides is a very effective means of antigen-specific immunotherapy. For self-antigens, it may not be informative if and when our proteins are expressed in the thymus, because thymic expression of these proteins will affect the developing T-cell repertoire. Nevertheless, treatment with soluble epitopes suppresses disease in relevant experimental models, and the treatment, most importantly, appears to be very well tolerated. It appears to be safe, with preliminary evidence of efficacy. Of course, the note of caution is that this is very early in the development of this approach, and we are planning a phase 2 trial to start as soon as possible. So with that, I'd like to thank those people who've been involved in all of this work. This is my group in Bristol. Bronwyn, Burton and Leona in particular helped define the mechanism of peptide immunotherapy in the animal models. Kirsty Nicholson helped initially with that project, and then was the first to work on the Factor VIII project. The initial clinical trial was run by Neil Scolding and Harry Pillai, and Heather Streeter did all of the immunology relating to that trial. Will Sanderson did some of the work on the Factor VIII design and the results that I showed you here, and the team at Epitope took forward the MS1467 into a clinical trial. This recent clinical trial, the PI was Jeremy Chataway at Queen Square in London. David Miller analysed all of the MRI activity for us. And I'd like to thank particularly Alla Zuzulia for providing the slide on the Lewis rat analysis that I showed you. And with that, I'll thank you very much.
Thank you very much, David. That was great, very interesting. I'd like to open the floor for questions, so please just raise your hands, and Bonnie, you want to go first? Just press it once.
Is that working?
Yes.
In your clinical trials, did you do any other types of measurements to see if the hypothesis you were proposing was actually working out, or if there was some other mechanism like blocking binding of the antigenic peptide or something like that?
Well, in the first trial, we have all the data from the first trial, and what I showed you was the immune response to myelin basic protein. At the same time, we were able to measure interleukin 10 by doing real-time PCR analysis. IL-10 is very difficult to measure because, of course, monocytes secrete IL-10. But we found that if you do the kinetics of IL-10 RNA expression and measure it by real-time PCR, that is the most effective way of measuring it. And in doing that, we focused in on the patient who was HLA-DR2 positive, and that patient showed an increase in IL-10 during the course of the treatment in response to myelin basic protein in vitro. So the T-cells stimulated with myelin basic protein in vitro, their level of RNA increased in a linear fashion throughout the course of the treatment, and then when it stopped, it went back down again. But that's only one individual, and we're awaiting the results from this latest clinical trial. I mean, the MRI data that I showed you is really news. Okay? Yeah.
Fanny, can you get it?
Still on?
Okay.
So I was struck by the fact that you're going to need to give the peptide basically continuously forever. So I wonder whether you've looked at transdermal patches. Just the route of administration, it seems like that would be a natural for something like that.
Yeah, and in fact, there's a group in Poland, Chris Selmai, and Chris's group have done exactly that. They've taken a peptide that is not a million miles dissimilar to the MS7 peptide that I talked about, and they've also included one peptide from myelin oligodendrocyte glycoprotein and the well-known SJL mouse T-cell epitope from proteolipid protein. They've put that onto a patch, and they've had actually very promising results, I would say, by using patches in treating MS. So it's looking promising, yeah. And I think that clearly is something to look at in the future.
I'm not sure if this is working or not.
It is,
That was, that was the foundation of my question as well. Because peptides delivered systemically typically have incredibly short half-life.
That's right.
It's possible that your peptides are not hanging around for long enough to really get to the dendritic cells or whatever to have the sustained impact that's needed. So have you looked at the PK actually?
We have, Valerie. I mean, we've done PK, functional PK, real PK, using the old intranasal route that we used to use, and then more functional PK by the subcutaneous route. And the truth is, you're right, but you're also wrong, if you'll excuse the expression. You're right in that if you take radioactive peptides and administer those, they're gone within 3 hours, right? However, if you then look at antigen-presenting cells, as far away from the site of injection as the spleen, for example, you will find functionally active presence of the peptide in terms of dendritic cells holding onto the peptide and expressing it for up to 5 days. Okay? And you'll notice that our treatment approach, the optimal treatment approach, is giving the peptide about once a week. Now, it is interesting actually about this continued dosing, because Harold von Boehmer here in Boston has tried giving peptides from pumps, from osmotic pumps. And if you administer a very, very, very low dose, but continuously for weeks at a time, what he found was induction of FOXP3 positive induced T regulatory cells. And I think the jury is currently out as to whether those cells, first of all, are stable, and secondly, whether they are functionally effective in an ongoing inflammatory environment. Whereas I think the mechanism that I described to you clearly is.
Lotte.
You described very nicely linked and bystander suppression. I was just wondering, where is the location of action of these IL-10 cells or producing cells, and how is that difference between self-antigens and foreign antigens?
Well, certainly I think, Lotte, for the mechanism that I described to you in preventing priming of an immune response, clearly I think that the action is around the dendritic cell, okay? Because the dendritic cell is the most potent cell in kicking off an immune response. There is no reason, however, to suspect that that phenomenon couldn't also apply to B cell antigen presentation. And I think B cells are often ignored by T-cell immunologists because they forget how important B cells are at amplifying immune responses. And I could imagine that this negative feedback mechanism would apply to that process as well.
Hi, David. Very nice talk. I'd like actually, speaking of B cells, and being the B cell guy in the audience here, to talk a little bit about the factor VIII studies. I recognize they're pretty early on, it looks like, but one might imagine. Well, first of all, are those antibodies somatically mutated and class switched?
Yes,
You know?
yes.
So that would imply these things have been through a dermal center reaction, right? And so my suspicion is that the mechanism of action there for this peptide approach is not going to be the same as what it is in things like MS.
Yeah.
And so it might involve blocking TFH production or formation, and maybe the B-cell presenter is actually more key to that phenomenon than in the MS studies, for example. So would you like to speculate on that for me?
Very much so. I mean, I think the mechanisms are going to be different, and I think what you'll have noticed is, of course, that this was starting treatment prior to immunization, which in many cases is not going to be a straightforward thing to do. However, I think there we are, we have to consider TFH germinal center formation, all those processes as a mechanism, but we haven't yet investigated any of that. And I think it will be very exciting to do so.
Okay, one final question.
Yeah, I was wondering if you compare what you've seen so far in your clinical study with what happens when you treat with Copaxone, which is basically a scrambled peptide approach. Would you see a reduced PBMC response to mild-basic protein? It's what I would expect, but I'm not sure whether you must have looked into whether that kind of gives a similar outcome.
In the Copaxone case, what people have reported, we haven't measured that ourselves. The comparison I showed you with Copaxone was in an experimental animal model. We haven't looked at the effect of Copaxone ourselves. The reported studies stress that when you treat with Copaxone, you induce a Th2 response to the drug, to Copaxone itself, and then some people find that emanates out into a Th2 response to myelin basic protein. Some people have noted switch from M1 to M2 macrophages, and others, I think Mike Racke in Texas, when he was in Texas, described induction of IL-10 secreting CD8 cells. So there's been a range of different mechanisms involved, but we haven't done a direct comparison ourselves, no.